Hello everyone, welcome back. So this is the revision class for the NPTEL course Experimental Biochemistry. This course is coordinated by Professor Das Gupta from the Department of Chemistry and Professor Dev from Department of Bioscience and Biotechnology, IIT Kharagpur. And I am Bias Mukherjee. And in today's class, we are going to go through all the topics that we have covered until now. We are going to go through MCQ questions only. So initially we'll start with the easy MCQs, okay, and then we'll go on to advanced level MCQs, and finally we will end with previous year's GATE 2023 to 2025 questions of both GATE XL and GATE BT, okay, and we will also go through a few of the questions from our previous class, that is week 11. So we'll go through some of those questions as well, because a lot of you found week 11 very difficult, and so we will go through those questions to help you out. Okay, so let's start with today's class. So it's only question answers, and the topics that we are going to cover are pH, molarity and buffer concept, which we had learned in our first class. Then we will talk about pKa, isoelectric point of amino acids, basics of amino acid and basic of protein and nucleic acids, enzyme kinetics and chromatography and its types, all the different biophysical techniques that we have covered in this course until today. And then we'll talk about purifications of nucleic acids and proteins, recombinant DNA technology. And in the last class, we talked about protein ligand interactions. So we'll go through those as well. Okay, protein ligand interaction, all the questions, few of the difficult questions basically. So let's start with the revision questions. So this is moderate level difficulty, and we'll start with the question. So the first question is, what is the molarity of a solution formed by mixing 100 ml of 0.5 molar HCl with 200 ml of 1 molar NaOH? Right, so we will look at the solution. So how are you going to solve the question? It's explained here for your understanding. The molarity of a solution is given by the moles of solute divided by the volume of the solution in liters, right? So that is the definition for molarity. Now, moles of HCl is equal to 0.05 moles, and moles of NaOH is 1 molar into 0.2 liters. So that is your 0.20 moles. Now, excess moles of NaOH are there by 0.15 moles. So the total volume of the final solution would become 300 ml, that is 0.3 liters. And the molarity is total number of moles by total volume. So that is 0.15 moles divided by 0.3 liter, and that gives us 0.5 molar. So the correct answer would be option number B, 0.5 molar. Okay, all right. So now the next question is, which amino acid would have the highest pi or isoelectric point? So glutamic acid, histidine, lysine and aspartic acid. So how do you calculate the pi? So the first amino acid would definitely not have the highest pi because it is an acidic amino acid, right? So it is going to have low pi because it is acidic. What about histidine? So if you look at all the pKa values, you will see that for lysine, the pKa value is maximum. And how do you calculate the pi? So we have talked about this in our third week's class, right? So how do you do that? So basically, you will have to take the two pKa values that are closest to the pH at which the net charge on the amino acid is 0, right? So in this case, that is pKa2 and pKa3, and so you take the average of these two and you get the highest pi. So for all the different amino acids, you will have to do the same, and then from there you will get whichever gives you the highest pi. Right, for the next question, in a denaturation experiment, a protein's secondary and tertiary structures are disrupted while its primary structure remains intact. Which of the following treatments is most likely responsible for this denaturation process? Now we have read about something like this in our previous experiments. For example, if you remember SDS page, there heat denaturation was done so that all the, actually give me 1 second, I'll be back. Okay. Yes. So if you remember that in heat denaturation, a lot of different bonds in the protein were broken, but the covalent bonds were intact. So that is, the primary structure was intact, but hydrogen bonds, hydrophobic interaction, etc., those were all broken. Right? What else? There can be BME treatment done to break the disulfide linkages. There can be treatment with any kind of chemical. So let's look at the different options. Exposure to extreme pH conditions, heating a protein solution to 100 degrees centigrade, and then adding a reducing agent such as beta-mercaptoethanol, treatment with a cheotropic agent such as urea, guanidine hydrochloride, etc. Right, so which of these would be responsible for totally breaking the secondary and tertiary structure while the primary structure remains intact? Okay, so let's look at the right answer. The right answer is C for the first question, and for the second one, cheotropic agents disrupt the protein structure by interfering with the hydrophobic interactions and hydrogen bonds. This disruption leads to denaturation of the protein's secondary and tertiary structure, leaving the primary structure intact. Now, besides this, we also can say that heating to 100 degrees centigrade can also disrupt the proteins secondary and tertiary structures. Okay, adding reducing agents such as beta-mercaptoethanol, this is also important for breaking the disulfide linkages, and extreme pH conditions can also disrupt the protein structure. Okay, so now we will move on to the next question. Okay, so which of the following amino acids is most likely to be found in the hydrophobic core of the protein? Now, in the hydrophobic core of the protein, it is mostly the hydrophobic amino acids will be there. So which of these are hydrophobic, do you think? So there is serine, there is lysine, there is tryptophan, and there is glutamine. Right, so let us look at the right answer. And the right answer is tryptophan, because tryptophan is large hydrophobic amino acid with a very bulky indole ring. It is commonly found buried within the hydrophobic core of the protein, where it contributes to stabilizing the protein's tertiary structure through hydrophobic interactions. Okay. The others have charged groups in their side chain, and therefore it is more preferred that they will be towards the surface of the protein. They will interact with other residues and with water to help with protein solubilization. Okay. Now, in recombinant DNA technology, plasmids are often used as vector. Which of the following is not a characteristic feature of a plasmid? Circular DNA, self-replicating, presence of introns, and antibiotic resistance genes. Okay, you know the right answer. Now, plasmids, the characteristic of a plasmid is CCC, that is covalently closed and circular, and it is supercoiled, right? So that means it is definitely circular DNA. It has to be self-replicating. It should contain an ORI site, right? And antibiotic resistance gene is one of the major properties. So the plasmid must give certain properties, certain advantages to the bacteria which is not present in its genome otherwise, so that this plasmid is held on by the bacteria in its body. Otherwise, the bacteria, if it does not require something, and then the bacteria will simply not carry the plasmid, right? In the next few generations, the plasmid will be lost. So that means definitely all of these are required except presence of introns. What about the next one? The process of introducing recombinant DNA into the host cell is, okay, what is it? Translation, transcription, transformation, and transduction. It is transformation if the host cell is bacteria. But if it is done using virus, then the process is transduction. Okay, now we will look at the right answers. So the right answers are presence of introns and transformation. So uptake of foreign DNA into the cell is known as transformation. Okay. If it is mediated by a virus, then it is transduction. Okay. Revision question, another one is there. So a biochemist is investigating the kinetics of an enzyme catalyzed reaction. Which of the following techniques would be most suitable for measuring the initial rate of the reaction under varying substrate concentration? Okay, so initial rate of the reaction. So gel electrophoresis, surface plasmon resonance, spectroscopy, ITC, and spectrophotometric assay coupled with enzyme kinetics analysis. So let's look at the right answer. The right answer is the fourth one, that is specific spectrophotometric analysis coupled with enzyme kinetics is widely used as a technique for studying enzyme kinetics. By measuring changes in the absorbance over time, the initial rate of the enzyme catalyzed reaction can be determined under varying substrate conditions, allowing for the determination of kinetic parameters such as Michaelis-Menten constants and maximum reaction rates, that is Dmax. Okay. Now we have two more questions. Which of the following is not a step in Sanger sequencing method? Annealing of the primers, DNA polymerization, denaturation of DNA, and amplification of DNA. So which of these do not happen in Sanger sequencing? Okay. Now, what is the purpose of using selectable markers in recombinant DNA technology? To identify recombinant DNA molecules, to visualize DNA fragments, to screen for the transformed cells, and to quantify the DNA amplification. Right? So we know about selectable markers. What do selectable markers help us with? For example, kanamycin is a selectable marker. Right? So it helps to select the transferments versus non-transferments. Correct? So then we will look at the right answers. In Sanger sequencing, if you see that Sanger sequencing involves DNA denaturation, primer annealing, DNA polymerization, and chain termination using DDNTPs, but not amplification. Okay. The next one is amplification is there in PCR, polymerase chain reaction. Okay. The next one is selectable markers such as antibiotic resistance gene are used to identify and select cells that have successfully taken up and expressed recombinant DNA. Right? The one that we were talking about. Okay. Now we have another question. In high performance liquid chromatography, that is HPLC, which detector is commonly used for detecting non-UV absorbing compounds? Okay. So what kind of detector? Refractive index detector, fluorescence detector, mass spectrometer, and electrochemical detector. So which detector do you think would be the right answer? It is mass spectrometer. So mass spectrometry is commonly used detector in HPLC for compounds that do not absorb UV light, providing highly sensitive detection and identification based on mass to charge ratios. Okay. Now, revision questions, advanced level. Okay, so these are questions from our previous class, that is week 11, protein ligand interactions, right? So the total heat released in ITC is product of individual heats, sum of the heats from each injection. So if you remember, there was a question where a graph was given, a thermograph was given, and there were these different peaks, and you had to tell whether the total heat released or absorbed was summation of all of these or multiplication or exactly what, right? So average of heat per injection, ratio of the heats. So that is a question that is actually repeated. Now the next one is, upon ligand binding to a protein, anisotropy decreases due to faster rotation, increases due to slower rotation, remains constant and becomes zero. So if you remember the formula for anisotropy, which we talked about in our previous class, you would be able to answer this question, right? So a sensogram shows fast association and very slow dissociation. What does this indicate? Weak binding. So if you remember, in SPR there was this association curve and then there was this dissociation and then it went down and then we had to recharge it again, right? So that was the curve in SPR. So the graph is called sensogram, right? So there is very fast association and then very slow dissociation. So what does that tell you? So that means the K on rate is high, K off rate is low. So what does that tell you? So weak binding, strong binding, that is low KD, no binding, and irreversible binding. Okay, so which of these would be true? So if you remember the formula for KD, it was K off by K on. So then what would this answer be? Correct? The next one is adding excess unlabeled DNA reduces shifted band. This confirms. So this is a question for IMSA. Right? So when you are adding unlabeled DNA into IMSA, that is electrophoretic mobility shift assay, why are you adding unlabeled DNA? To ensure that there is binding between the labeled DNA and the protein. So there is this competitive assay that is going on, right? So this confirms non-specific binding, specific binding. So what do we see? Adding excess unlabeled DNA reduces the shifted band. Okay, specific binding, irreversible binding, and covalent binding. So exactly what is happening? Let's look at the right answers. Okay, so we see that Q total, the net amount of heat that is released, it is always the summation of all the peaks that are there. Okay, so all the peaks are there, and if it is an exothermic reaction, they will be downward. If it is an endothermic reaction, the peaks will be upward, and it is always the summation of all the peaks. Okay, now for the next question, anisotropy increases due to slower rotation. Okay, the next one is, of course, strong binding, that is low KD. Why? Because KD is equal to K off by K on. If the K off is less and the K on is very high, then KD will be low. Correct? And that would indicate strong binding. The next one, adding excess unlabeled DNA reduces the shifted band. So that tells you that the binding is very, very specific. So to confirm the specificity of binding, this process is actually performed. EMSA with competitive EMSA is actually performed. Correct? Now we have few more questions. A competitor increases the apparent KD. This means binding affinity increases, binding affinity decreases, no effect, protein denatures. So if you remember about this enzyme competitor inhibitor, then you will be able to answer this question. If binding is enthalpy driven, which is true, delta H is more than 0, delta H is less than 0, delta S dominates, and there is no heat change. Okay, this is a question from ITC. If Hill coefficient N equals to 1, then there is negative cooperativity, no cooperativity, positive cooperativity, and competitive inhibition. Now we have not talked about Hill coefficient. So that is why I have added 2 slides on Hill coefficient right after this slide. Okay. Now in an ITC experiment, the peak decreases in magnitude with each injection and eventually flattens. This is something that we know, right? Why is that happening? Okay, and which of the following options is true? So ligand concentration is limiting, protein becomes saturated over time, heat of the dilution dominates from the beginning, binding affinity increases over injections. Okay, let's look at the right answer. So the first one is competitor increases apparent KD, so binding affinity decreases, definitely, because we know that for competitive inhibitor, it is going to bind at the active site and it is going to prevent the substrate from coming and binding, right? So the ligand is not able to bind because the competitor is sitting there. So its binding affinity obviously decreased. The next one, if binding is enthalpy driven, then enthalpy will be negative, right? So enthalpy is less than 0. Okay. Now, Hill coefficient, if it is more than 1, then it is positive cooperativity. Correct? The last one is protein becomes saturated over time, and that is why all the peaks finally reduce and there is the baseline. Right? So now we will see what exactly is Hill coefficient. Okay? So Hill coefficient is a quantitative measure of the degree of cooperativity in a system. It is derived from the Hill equation, which describes the binding of a ligand to multi-subunit protein or enzyme. The Hill coefficient can be interpreted as NH equals to 1, indicates non-cooperative binding. So each ligand binds independently as seen in enzymes with single binding site, that is non-allosteric enzymes, right? Or when the subunit does not influence each other. Okay, now NH is more than 1. Then it is positive cooperativity in enzyme ligand binding. When one ligand binds to a site and it increases the likelihood that additional ligands will also come and bind to other sites. The substrate binding curve for this type of enzyme appear as S-shaped curve. Now you remember the allosteric curve for allosteric enzymes, right? It is S-shaped. As the binding of the ligands initially increases, the rate of ligand binding. So if you remember hemoglobin, cooperativity is seen there too, right? Now, NH is less than 1. That means there is negative cooperativity. So in enzyme ligand system, in this case, binding of one ligand decreases the affinity of the enzyme for subsequent ligands, making it harder for them to bind. So negative cooperativity makes tends to flatten the substrate binding curve. Okay. Now this is the graph. You see that as initially it is just flat, but as you keep on increasing n, it becomes more and more S-shaped. Right? So the Hill coefficient provides insights into the regulatory mechanisms of enzymes and proteins, particularly in complex biological systems. Positive cooperativity is crucial for the efficient function of enzyme like hemoglobin, allowing rapid responses to change in ligand concentrations. Conversely, negative cooperativity can fine-tune enzyme activity, preventing excessive substrate binding. Okay, now we have more questions. What will be the change in pH when 0.02 moles of HCl is added to 1 liter of buffer solution containing 0.1 molar ammonia and 0.1 molar NH4Cl? pKa of NH4 plus is 9.25, right? So let's see how to solve this particular question. Okay, so to find the change in pH, we will use the Henderson-Hasselbalch equation. So pH, you know, is equal to pKa plus log of A minus by HA, which is Henderson-Hasselbalch equation. And then initially the concentrations are given. pKa is equal to 9.25. So pH becomes pKa plus log of 0.1 by 0.1, that is 9.25, right? Okay. Now, when 0.02 moles of HCl are added, it reacts with NH3 to form NH4 plus. So the equation is NH3 plus HCl gives NH4 plus Cl minus. The change in the concentration of ammonia and NH4 plus is equal to the moles of HCl added, which is 0.02 moles. Now concentration of ammonia is 0.1 minus 0.02, that is 0.08 molar. Concentration of NH4 plus is 0.1 plus 0.02, that is 0.12 molar, and pH is equal to 9.25 plus log of 0.12 divided by 0.08, that is more or less equal to 9.72. So the change in pH would be 9.72 minus 9.25, that is 0.74, and the final answer would be increase by 0.47. Right, so 0.47, and so it would be increased by 0.47. Okay, now we'll look at the next question. So what is the approximate PI of a dipeptide composed of glycine and alanine? Okay, now it's a dipeptide made up of glycine and alanine. Both of them are uncharged. So how do you think the PI would look like? Okay, let's look at how to solve this. To find the PI of the dipeptide, we take the average of the pKa values closest to the point where it carries no net charge. The PI of the dipeptide would be average of the pKa values for the amino acid it contains. For glycine and alanine, the pKa values are 2.4 and 9.6 respectively. And so the average pKa would be 2.4 plus 9.6 divided by 2, that is 6. Therefore, the correct answer is B, and it is 6. Okay, so all of the values are given to you and you would be able to find out the right answers from here. It is very simple. Okay, now we go to the next questions. What method is suitable for determining the secondary structure content of a protein? This you know. Okay, there is ITC, there is DLS, there is CD spectroscopy and mass spectrometry. The next question is, which technique is utilized to study the dynamic and conformational changes in a protein? Okay, right. So CD spectroscopy is what would tell you about the secondary structure of a protein. What about dynamics and conformational changes? Of course, NMR is one of the most famous techniques for this. You can also use FRET. FRET is fluorescence resonance energy transfer. So that can also be used. Okay, right. Now, we have two more questions. Which technique is most suitable for studying the kinetics of protein folding and unfolding transitions? What method is most suitable for determining the stoichiometry of a protein complex? This also you know. So there is analytical ultracentrifuge, there is ACC, there is ITC, and there is NMR spectroscopy. Which of these would be used? Right. So we will look at the right answers. So the right answer is both CD and FRET for the first one, and it is ITC. So stoichiometry of protein complexes, you can also use analytical ultracentrifuge for that. Okay, and also ACC and ITC. But yeah, ITC would give you very right answer for that matter. Okay. So which of the following is a potential ethical concern associated with recombinant DNA technology? And which is a potential concern, do you think? Loss of genetic diversity, increased risk of genetic disease, patenting of genes, environmental pollution. Which of the following factors does not affect the retention time of a compound in chromatography? Column length, mobile phase composition, detector sensitivity, and sample concentration. Let's see. So, patenting of genes is definitely not a potential ethical concern associated with recombinant DNA technology. All of the others would be. Okay. Now, so basically patenting of genes is a potential ethical concern concerning RDT. The others are not. Okay. So the patenting of gene raises ethical concerns related to the ownership and control of the genetic information, potentially limiting access to important technologies and discoveries. Which of the following factors does not affect the retention time of a compound? And that would be detector sensitivity, because all of the other factors influence this. So detector sensitivity does not directly affect the retention time of a compound. In the chromatography, retention time is primarily influenced by column length, mobile phase composition, and sample concentration. Okay. Now, so now we have gate BT and gate XL PYQs. Okay. So we will go through them quickly. Right. So this is a question from GATE BT 2023. In hybridoma technology, which one of the following enzymes is absent in the myeloma cells that are used for monoclonal antibody preparation? The options for you are AGPRT, then you have alanine aminotransferase, and triose phosphate isomerase, and glycoside transferase. Now we have talked about this, so I'm sure you'll be able to answer. Now, which one of the following methods cannot be used to determine secondary structure of a protein? CD spectroscopy, Fourier transform, IR spectroscopy, and mass spectrometry, and X-ray crystallography. Very straightforward. Let's try to look at the right answer. Okay. So, okay. Now we have AGPRT would be absent in the myeloma cells, okay, in monoclonal antibody production. Now, which cannot be used to determine the secondary structure? Of course, CD can determine the secondary structure. What about the X-ray crystallography? You can still know if it is a unfolded protein or a folded protein, right? And what about the FTIR? That can also tell you, but mass spectroscopy is not going to tell you whether it is folded or unfolded, whether it's an alpha helix or a beta sheet. It is only going to tell you the sequence of the amino acids in that protein. Okay, now we have 3 NATs, numerical aptitude tests. Okay, we will try to look at the answers of each of these. So the first question is, aqueous two-phase extraction is used to recover alpha amylase enzyme from solution. A polypropylene glycol dextran mixture is added to the solution and it separates into upper and lower phases, right? So the partition coefficient is 4 and the ratio of upper to lower phase is 5. Upper to lower phase volume is 5. The enzyme recovery or yield in percentage rounded off to the nearest integer is. So we will look at how to solve this, okay? Right, so the solution is given to you and the answer to the question is also given to you, okay? So the answer is 94 to 96 percentage, okay? Now, aqueous two-phase extraction. So how are they doing it? So you know that partition coefficient is equal to C upper by C lower and it is said that partition coefficient is 4. What about volume ratio? V upper by V lower is 5. That is given. Right. Now recovery is equal to partition coefficient into V upper divided by partition coefficient into V upper plus V lower. Right. So what you do is 4 into 5 divided by 4 into 5 plus 1. So that is 20 by 21 and you calculate the percentage and you get 95.24 percentage. Okay. That is the enzyme yield. Now the next question. The number of different possible ways of forming 5 intramolecular disulfide bonds with 10 cysteine residues of a protein is. How do you do it? The formula is we need to count the number of ways to form 5 disulfide bonds from 10 cysteine residues at the number of ways to divide 10 items into 5 unordered pair, that is 10 factorial divided by 2 to the power 5 into 5 factorial. So 10 factorial is arrange all cysteines to the power 5, which each pair can be arranged in 2 ways. So avoid double counting and 5 prime is the order of pairs, doesn't matter. Okay, so now you get this full value and then when you multiply you get 945. So the right answer is 945 to be exact. Okay, and there were 2 marks for this particular question. What about the next question? A DNTP master mix is prepared by combining 40 microliter of each 20 millimolar DNTP stock. So each of DATPs, DCTP, DGTP and DTTP was taken. 4 microliter of this DNTP mixture is added to a PCR mix and the final volume is adjusted to 50 microliter, the concentration in micromolar of total DNTPs in the PCR mix is, right? So the right answer is 1600 micromolar. How is it coming to be 1600 micromolar? Let's see. So 40 microliter of each of the 4 DNTPs. So the total volume is 160 microliter. So each stock contains 20 millimolar. So each DNTP is diluted to 5 millimolar. Correct. So the master mix contains 5 millimolar of each NTP. So then the total concentration would be 5 into 4, that is 20 millimolar. Now 4 microliter of the master mix is mixed into 50 microliter of total volume. So the dilution factor is 0.08. Right. So 4 divided by 50 becomes 0.08 and the final concentration is 20 millimolar into 0.08. That gives you 1.6 millimolar and that into micromolar is 1600 micromolar. Okay, great. Now we have 2 more questions. One of them is from bioinformatics. So which of the following programs is used for finding distantly related or remote protein homologs? BLAST10, BLASTX, SciBLAST and you have TBLASTX. Okay. And the next one is direct DNA transfer methods used for plant genetic engineering is or are microparticle bombardment, electroporation, polyethylene glycol treatment, and agrobacterium tumefaciens mediated transfer. Agrobacterium mediated transformation. Okay. Which one do you think would be the right answer? So the right answer is PSI blast or PSI-blast for the first one, and it is microparticle bombardment, electroporation, and polyethylene glycol treatment, all the three, because agrobacterium mediated method is indirect method. It requires a living vector, that is a bacterium, and DNA transfer happens by a natural infection process and not direct insertion. Okay. Right. Now, which one of the following restriction endonucleases is a blunt cutter? Okay, so BAM H1, EcoR1, HIN3, all of these are sticky-ended restriction enzymes, but EcoRB is not sticky-ended, it is a blunt cutter. Okay. The next one is, which one of the following peptides do or does not absorb ultraviolet light above 250 nanometer wavelength? So all the different peptides are given. Let's look at them. Okay, so the right answer is that peptide bond absorbs at 190 to 220 nanometer. Phenylalanine absorbs at 257 nanometer. Cysteine is 250 to 260 nanometer. Tyrosine is 275 nanometer. Tryptophan is 280 nanometer. So absorbance above 250, that is 260 to 280 nanometer in peptides, mainly comes from aromatic regions, right? So tryptophan has the strongest absorption, tyrosine moderate 275, phenylalanine is 257 nanometer, but still more than 250. So peptides without the aromatic residue do not absorb above 250 nanometer. So you can see that the last two peptides, they do not have any aromatic residue, so they are the ones that do not absorb above 250 nanometers. Okay, now we have a very nice question, very interesting question. So E. coli is grown exclusively in a medium containing 15N NH4Cl as the sole nitrogen source, and subsequently the cells were shifted to a medium containing N14 NH4Cl. The molar ratio of the hybrid N15 to N14 to light DNA, that is N14 after 4 generations rounded up to 2 decimal will be. Okay, so this has been taken from the Michelson-Stahl experiment, and you know about the Michelson-Stahl, I'm sure. So the experiment is explained here for you. Okay, right? So initially there is this 15N DNA, and then in generation 1 there are 2 copies of 15N DNA and 2 copies of 14N DNA, right? And then in generation 2 there are what? They are in the ratio 1:4, because there are 2 copies of 15N DNA, but 1, 2, 3, 4, 5, 6, right? So 6 different copies of 14N DNA. So 1:3 ratio, correct? 1:3 ratio, and then when you spin it, the heavy DNA or the 15N DNA is at 1.14, 15, that is mid-sized DNA is there, and 14 and 49 is light DNA, right? So all of these different DNA are there. Now how do we solve this question? So initially all DNA is heavy DNA, so that is 15-15. Now, after shifting to 14N media, newly synthesized strand contains 14N DNA. In semi-conservative replication, each daughter DNA has one parental strand and one new strand. So you can see the table is given. In generation 0, it is only 15N 15N DNA, heavy DNA. In the first generation, it's a hybrid. In the next generation, there are 2 hybrids and 2 light chains. In the next generation, there are once again 2 hybrids and there are 6 light chains. In the next generation, there are 2 hybrids and 14 light chains. So the light chains keep on increasing, right? So the 2 original heavy strands always remain part of the hybrid DNA, and therefore the hybrid molecules remain constant, that is 2. Right, so the total DNA doubles after every generation. So after 4th generation, it is 2 to the power 4, so that is 16. Now total number of light chains is 16 minus 2, that is 14 hen. So the total after 4 generations, the total DNA molecules is 2 to the power 4, that is 16. So number of hybrid DNA chains are 2, and number of light DNA chains are 16 minus 2, that is 14. Hybrid is to light is 2 divided by 14, that is 1 by 7, and 1 by 7, if you convert it, it becomes 0.14. Right, so the right answer would be 0.14. Give me a second, I will be back. Okay. Okay, so I'm back. Now we look at the next question. Okay, so the next question is very interesting. Let's look at this. A culture of Lactobacillus Escherichia coli is grown in medium lacking lactose or any other beta-galactoside. Okay, the response of the lac operon upon induction by lactose can be monitored by measuring the levels of lac mRNA, beta-galactosidase enzyme, and permease enzyme, which are the products, right? Which one of the following profiles correctly captures the on-off response to lactose? So there are 4 different profiles given, PQRS. Now you will have to select. Okay. So how do we solve it? Right. So see, lac mRNA is produced rapidly after lactose is added, degraded quickly once lactose is removed, and the proteins beta-galactosidase and permease, they appear after a delay. So translation obviously takes time. They persist longer, so the proteins are more stable than mRNA, correct? So what should be the trend? The trend should be in this way, that the mRNA rises first and falls fastest. Proteins rise later. Protein declines slowly after lactose removal. So then which one is correct? P would be the right answer. Okay. Now, the human genetic code is triplet in nature with 64 codons made using 4 nucleotides. If the human genetic code was doublet in nature, the number of codons theoretically possible from 4 nucleotide is, okay, answer in integer. If the genetic code was doublet instead of a triplet, that is 2 nucleotides per codon, there are 4 different nucleotides, then each position of the codon can be filled with 4 different ways. So the total combination would be 4 into 4, that is 4 to the power 2, and 4 to the power 2 is equal to 16. So then the number of codons theoretically possible would become 16 instead of 64. So from where is 64 coming? 4 to the power 3 is 64, correct? That is why the total number of codons is 64. Okay, the spectrum of a protein obtained using electrospray ionization mass spectrometry, ESIMS. This is a question from GATE XL 2024. Okay, so there we have covered questions from GATE BT 2023, GATE XL 2025, and GATE XL 2024. Okay, let's look at this one. So the SIMS is shown below. 2 peaks, one at M by Z equals to 2960.6 and the other at M by Z 3552.5 are marked. The mass of the protein associated with the M by Z is 2960.6. Peak is dash Dalton. Okay, so the relative abundance is given, M by Z ratio is given. So what is going on here? Now they have asked you to calculate the mass. So how are you going to calculate? The ESIMS proteins appear as multiply charged ions, and the relation is M by Z is equal to capital M plus Z into H divided by Z. Okay, and where capital M is equal to molecular mass of the protein, Z you know it is the charge, and H is equal to 1 Dalton, that is mass of the proton. Correct. Now given M by Z1 is equal to 2.9 Dalton, 2.9 kDa, and M by Z2 that is 3.5 kDa. In ESI spectra, adjacent peaks differ by 1 charge. So assume 2.6 is equal to M by Z, M by Z by Z, and 3.5 is M plus Z minus 1 divided by Z minus 1. So then you know that if you solve this, you will get that this when you equate both of these equations, you get M is equal to 17.5 kDa. Okay, so that becomes the mass. So M by Z becomes 17.4 kDa. So for reference, I have taken questions from these two websites majorly, but all the other questions were also taken from different websites. So you can practice those questions. Okay, I'll update this reference slide before uploading it. Now we will go to another slide. Okay, we will go to another. Yeah, so we'll go to this one, lecture 11. Because we need to discuss the questions where you guys had doubt. So we have this lecture 11, and we will go to the questions in which you guys had some doubts. Okay. So first is this question. I will remove this. Yeah. So there is this particular enzyme. It binds with a newly synthesized drug molecule with an association constant Ka. What is likely measured Kd in the presence of a competitive inhibitor? Now you know that in the presence of a competitive inhibitor, the Kd must decrease. And if it is decreasing, that means it is becoming what? Kd would be, so Kd here in this case, it would be 1 divided by 10 to the power 7. So that makes it 10 to the power minus 7. Right? 10 to the power minus 7 mole inverse. And then what happens is, and then it should decrease. So if it decreases, that means it is going to become what? 10 to the power minus 6. Correct? So the right answer would be yeah. So in competitive inhibition, the inhibitor, so true Kd is and then it should be higher than this. So that means 1 minute. Yeah. So it should be this. This should be the right answer. Kd minus 6 should be the right answer because affinity is decreasing. Right? So in competitive inhibitor, so the measured Kd increases, that means weaker binding is there. True Kd is 10 to the power minus 7, but measured Kd is higher than this. Okay. Now the next one is, okay, there is no problem with the next one. We'll go to this particular question. Okay. Right. So this is a very interesting question. So let's see that K-on and K-off values for the ligands X and Y with the enzyme E is given below. Okay, and now they are asking you which of the following, whether X or Y, which one binds more tightly with E, and which complex lives longer. So how are you going to find that out? So the right answer is C, of course, but then how do you calculate, right? So the calculation is given here. So binding affinity is equal to K off by K on. So Kd we know that it is K off by K on. It is a measure of how tight binding the complex has. So for X it is 10 to the power minus 7. For Y it is 10 to the power minus 8. So that means Y is definitely smaller. That means the Y is binding tighter. Right? So Y binds more tightly. That is the conclusion. Now the next one, lifetime of the complex, you know that lifetime or tau is equal to 1 by k of. So then what do you see? That tau for x is equal to 1000 seconds, tau for y is 10 seconds. So XE complex lives longer. Okay. So Y binds tighter, but XE lives longer. So 2 and 3 are the right answers. Okay. Now we'll go to the next question. The next question is this one. The researchers designed a new drug and they want to study the interaction of this drug molecule with human peptidase enzyme. If the peptidase drug molecule complex dissociates with the off rate, that is k of 1549 second inverse, and what is the time constant or tau for this reaction? So how long does the complex live? Very similar to a previous question. So k of is given, then what would be tau? Tau is equal to 1 by k of. Correct? So if you just do 1 by this, you will get that tau is equal to 645.5 microseconds. Okay. Now fast dissociation means high k of and tau is small, that means it is weak binding. And slow dissociation means k of is low, tau is large, and there is strong binding. And this definitely follows first order kinetics. Now we go to the next question that you guys found difficult. Yeah, this one. So here, 1 minute. Right. So here what is happening? Wait. So the dissociation constant K of for a protein DNA interaction is determined by EMSA. The intensity of the free ligand determined at a corresponding time interval is given in the table below. Determine the K of. So intensity is given and time is given. So how do we calculate K of? Dissociation constant, you know, follows first order kinetics. So PL breaks into P plus L. The complex decay, you know that I is equal to I naught. So I is equal to I naught minus I naught into e to the power minus k of into t, right? So ln of PL is equal to minus k of into t plus c. C is the constant. So since free ligand increases towards the saturation, that is 1000 intensity, half-life occurs when the intensity is 500. So at time 5 and 10, you see that the intensity is 393 and 632. So that is nearly half. So half, that is nearly 500, lies between 5 and 10 minutes. Average is 7 minutes. Now if you put in the equation of half-life, you have T half is equal to ln 2 by K of, and K of comes to be 0.7 divided by 7, so that is nearly 0.1 minute. Okay. Now another question. Okay. So a very similar protein ligand titration experiment was repeated, where the protein concentration of 10 micromolar is used, which is more than the expected Kd. The concentration of the ligand and the protein ligand complex for each titration point is given below. Determine the dissociation constant Kd. So ligand, different values are given, protein ligand complex, different values are given. What do we want to find out? We want to find out the Kd. So how do we find out the Kd? So here this is a NAT, right? And the right answer is around 6 micromolar. So how is that coming? So let's look at the right answer. Protein concentration is 10 micromolar. Binding equation is PL is equal to P total into L divided by Kd plus L. At half saturation, PL is equal to P total by 2. So PL is equal to PT plus LT, that is total. Okay, PT is 10 micromolar, ligand total is total ligand concentration, plus Kd minus root of PT plus LT plus Kd whole square minus 4 into PT plus LT. So P total is 10 micromolar, half binding, that is PL is equal to 5 micromolar. From the table we see that ligand concentration is 6 when PL is 5 micromolar. So that means Kd will become 6 micromolar. Okay. And that's all. So we end today's class. Thank you for your attention. Thank you for your time. Okay, we'll meet again in the next week for our week 12 class, which would be our last class for this particular course this semester. Okay. All the best for your exams. I'll be ending the call now.